So we're going to get started. Our next speaker is Mark Ptashne, who's the Ludwig Chair of Molecular Biology at Memorial Sloan Kettering Cancer Center. So Mark is another giant of molecular biology who's made seminal contributions in a number of areas, particularly understanding how genes are regulated. Back in the 1960s, we had an idea about gene regulation based on sophisticated genetic experiments, but there was no biochemistry. So Mark undertook some audacious experiments with bacteriophage lambda, isolated the lambda repressor, and showed it bound DNA and repressed transcription. This was the first site-specific DNA binding protein. It was very exciting work. And he's continued to elucidate many of the central features of gene regulation and gene expression. He's also shared his insights in a book, The Genetic Switch, but his passion extends beyond the laboratory, and many of us in this room have had the pleasure of hearing Mark perform on his Stradivarius violin. So Mark met Sid when they were both working in Matt Meselson's laboratory and known each other for many years. So I believe that Sid knows Mark really well. So to introduce Mark, I will quote Sid, because Sid invited Mark several years ago, and he said, and I quote, "Our speaker today is Mark Ptashne, a notoriously smart guy." And that you can see that from the title of his talk. Mark?
Oh, can I be heard? Yes. That's it. No more blah, blah about Sid. I mean, enough is enough. So, so years ago, I went to my thesis advisor, Matthew Messelson, and I said, Matt, I just heard a completely weird talk by this guy who you think is a great guy. And Matt said, yeah, well, you don't understand how he operates. What he does is he sits at home and speaks and films a really clear, detailed, scientifically proper talk. Then he gets on stage, he puts it in his brain, gets on stage, clicks, and then he comments on it as it goes along. That's why it's a little odd. So Tom said, we drove up, he said, that's what you should do. So this talk is going to have some of that. A lot of it you already know or won't admit to not knowing, and so. Okay, so originally, I mean, the specificity. So why should we think about that? Well, one of the most galvanizing things about Jacob-Monod was specificity. There was a specific regulatory gene that regulated a specific target gene. So how does it work? So, of course, one possibility was RNA at either end. And it would have been sort of more commodious had it turned out that way. We could all have an RNA fest, but it didn't turn out that way. And although RNA is not completely irrelevant, in most cases, it's a protein. And of course, we immediately knew that the protein would bind to a specific site as defined by genetics as being important. And when all the huff and puff was over, we found out at heart what determines specificity. It's variations on this very simple principle that an alpha helix is in the major groove. It looks at the base pairs and so on. Now that's a very thin and unreliable read on which to base a whole regulatory system because there's not much specificity, there's very little binding energy. So we consult the Bible, which is this fantastic little book which nobody but me seems to have read, and very old systems biologists called The Sciences of the Artificial by Herbert Simon. If you don't get anything else out of this talk, go to Kindle and get it. 3rd edition. How to build a reliable system from unreliable parts. This is a standard engineering problem. And since he thought biology should be regarded as a design problem, just like everything else, and he says we can cope with the unreliability only by our manner of organizing these parts. So now you worry, this is a famously elaborate intellect mastering all kinds of fields and so on. So it's going to get pretty abstract pretty fast. No, he says the requirements, Herbert Simon, the requirements of design can be met fully by a modest adaptation. It's called Sciences of the Artificial. He kept up with Genetic Switch. He made one in the 60s. He made it in the 3rd edition just before he died in the 90s. So To our relief, he says the requirements of design can be met by a modest adaptation of ordinary declarative logic. Whew. So let's see how far that can get us. Well, that initial thing, of course. Sorry? Matt, are you going to interrupt or what? Okay. Sidney? So, how does it work? Well, the way it works is by just the dumbest way possible. You take the basic reaction, you reiterate, you invert, you blah, 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 and it starts to look like this. You make interactions between the proteins, and pretty soon you've increased the specificity a huge amount. Now, the important point here is that it depends on protein-DNA interactions, protein-protein interactions, and that, of course, when you get into the higher world of where everything is, can be very confusing because proteins can go from here to there depending on their binding partner. So the whole idea of cis-regulatory changes, yeah, they're important, but in yeast, we know a lot of stuff is not that. It has to do with the interaction of guys. And then the most scary thing is, of course, it depends on concentration, a matter I'll come back to. So this simple picture has a bonus. The bonus is that cooperativity explains, quote unquote, gene activation. How a regulator can turn on, as we say, transcription of a gene. What we have to do is just replace one of these dimers with RNA polymerase in E. coli, and we get this sort of picture. The polymerase is binding with a weak affinity to the promoter. The activator binds and then touches the polymerase, and that increases the rate of transcription. Now, at Cold Spring Harbor, I got into a big thing. The past is always repeated by the enthusiastic youth. There is, years of arguments because you can get a KB effect, a KD effect. Doesn't matter. In all these cases, we know from crystallography, from genetics, that any contact between the activator and the polymerase, when they're both at their positions, will do this thing called activate, namely make the polymerase come and transcribe more frequently. And as I say, the key experiments are that you can change these contacts. There's nothing special about a region that touches polymerase, just a couple of amino acids. It's the dumbest possible way to run the universe. And it turns out that's how it works in eukaryotes too, in a way even simpler because it's slightly, it really doesn't matter to a large extent. A matter I'll come back to. Let's see, how do you get a pointer out of this? Yeah, so this little guy here, who is a DNA binding domain of any type, attached to what's called an activating region, will activate, and the amazing thing is, it activates any gene in any organism as far as tested. So sometimes things are pre-bound and people don't understand this, and then it will recruit what you need to do the rest of it. Plants, Tom and I showed flies, plants, all kinds of stuff. And so this still is a slightly miraculous business, and it's not being studied very much. Steve Hahn is one of the people, and he knows it touches and apparently changes its configuration and so on and so forth. And somehow it triggers this assembly of 100 proteins or whatever it is, and you get transcription. And just remember one of Jacob-Monod's rules, the question is how you regulate transcription, not what happens as you transcribe. Because people, right? We'll come back to what we do about nucleosomes in a minute. And the important point is that the conceptual, the evidence for this is just as in bacteria. You can, many different activating regions, it's actually simpler. Many different activating regions work. If you put in artificial protein-protein contacts, it works. It's incredibly easy to get it to go, to work it. So evolution has no problem doing this. Moreover, as you might have guessed from these kinds of pictures, any activator can it works on just depends on, finally the word, specificity is meaning which gene is activated, just depends on the domain. So if you put the thing in front of one gene, do a first approximation, it activates that gene. Put it in front of another gene, it activates that gene. So evolution is working with basically a logo type set. It's almost embarrassingly simple. And for example, one of the things you can do is to put the activator next to its own gene, so you get a positive feedback loop. And in fact, that's how it works in lambda, to make a lysogen where this thing is making this thing, which is unfortunately called a repressor, but it's activating here. The cells divide, and this is incredibly stable, an incredibly stable differentiated state, if I may use that word. And just in case you might want to hear this, oh, there it is again, I guess. Because the cells are dividing, the repressor level, the repressor activator level can fluctuate. So to guard against that, you introduce negative feedback, which either increases the rate or decreases the rate as needed, a governor. So, but there's nothing fancy about this. There's another thing that Simon goes into. How would you calculate how to do this? Well, you can't calculate, but if you just fiddle around with a binding site and make it of the appropriate affinity, you will get this effect. People can interrupt me if they. And it turns out, much to one's pleasure, that this is the way it almost certainly, well, it certainly works in certain higher organisms and maybe all. So for example, here's Hobart's famous picture of a worm sensory neuron. Which was a gene, which is a transcriptional activator. It's got a binding site nearby, so it's turning itself on, and as it turns on these other guys. And the amazing fact is that if you interrupt this loop in many different ways, the differentiated state goes away. And there was a review citing, I don't know, there are lots of cases. So the mystical stuff about chromatin states and so on is even more silly than it used to be. Okay. Now, this is maintenance that I've just described, maintenance of the differentiated state. How do you get there? Well, the way you get there is by establishment, and that's a huff and a puff. That's where you get real. So, for example, in sea urchins, according to the late Eric, you get this kind of thing, which is like 1,000 lambdas all mixed up.
What is that circuit? Tell me what that is.
That is sea urchin. Well, see, I can tell by just looking at. Oh, it ends up making activators that then self-activate and maintain the differentiated state. That's what differentiation is. Getting the right activators that according to this view, which has evidence actually, you have to do this and make these different cell types, which will then make different autoactivators that perpetuate themselves. And here, you're not dividing because you're differentiated, so you don't need the negative feedback. Okay?
Glitches, there must be glitches, though, that end the process.
Well, man, most developmental processes don't work. I mean, most embryos don't make it.
It'd be nice to figure that out, wouldn't it?
No, because, well, what do you mean? I mean, there are too many ways to interfere. What I suggest to Matt, who always poo-poos it, is that the reason you age is that these positive feedback loops with a certain frequency get lost, and you can't get them back without going through some kind of process, right? So of course, you just blow it in pretty soon. So Matt will say, well, yeah, but what causes the loops to, anyway. In lambda, we used to think this was complicated. I don't want to go into the details because I'm going to get to the underlying point in a minute, but it's this sort of thing. But just so you can understand it, the phage comes in, it makes an activator that works like any old activator. It goes here and makes this thing called the repressor, which then feeds back and turns itself on, and these are off now, and so you have a self-maintaining state. So this is the equivalent of, from here to here is establishment, and the other thing is maintenance. Okay, so far the principles of everything that I've talked about, I would argue, are general, and they apply to bacteria and to eukaryotes, and surprisingly now, nucleosomes. Ta-da! So I don't want to, we spent 3 years on this or something, developing a new assay to tell how, you see, there was this big business where people would calculate fancy models for where nucleosomes were bound. And it was just all, in my opinion, totally irrelevant and misleading. It goes like this. Nucleosomes form at random But if you regard them as DNA binding proteins and measure their avidities with this special assay, you find that the higher the GC content, the tighter they are bound. Now, the non-intuitive aspect of this, actually, we don't even know why that's true. At least I don't. Roger, can you explain that? Anyway, the effect of the GCs. Good. Okay, so here's something I think Roger suggested years ago. Phasing, the precise positioning of nucleosomes, is a completely independent variable. It has to do with barriers. So if you have a barrier so that the nucleosomes can't move, it doesn't matter whether they're binding tightly or weakly. This is quite contrary to how typical DNA binding proteins work, right? The tighter they bind, the more they're going to be precisely positioned. With nucleosomes, it doesn't matter. They have blah, blah, blah. But if they run into a barrier, and I'll show you an example, they tend to phase. And everybody was trying to figure out from the sequence why they phase, so of course it was all hopelessly wrong. So let me tell you now what I think are 3 good news aspects of random nucleosome formation. First of all, of course, everyone, I think, agrees that it expands the ways you can do cooperativity. So if 2 guys compete for the same nucleosome, DNA in the same nucleosome, they'll, of course, help each other even if they don't touch. In bacteria, they have to touch. But here you've got, right? And there are variations on this. One guy can have an active, anyway, that doesn't matter. But that's good. And that partly explains why eukaryotes seem like such a mess. I mean, you get these guys who are binding cooperatively, all over the place. So, whoop, I didn't know. Wow, I had no idea that was going to happen. Okay. Now here's something I'll never convince people of. The great news about, even though sometimes people say it, they don't understand. Okay. You don't need, in eukaryotes, promoter-occluding repressors. Let me give you an example of why you do need them, or why you need them in bacteria. In bacteria, this recruiting reaction is, of course, it's a factor of 10, whatever it is. And this, if you take away the activator, the polymerase will give you a significant basal level of expression. The only way you counter that basal level is by adding a repressor. And in fact, to my knowledge, all activators that work this way in bacteria are paired with repressors. So there's something called lambda repressor working as an activator in CRO. There's CAP and LAC repressor, and on and on. So the idea is that when you get these huge factors of induction just using binding interactions, which either push up the basal level or push it down. And as you know, Jacob-Monod just doesn't really matter, but the only thing they knew about were the repressors, and so it's another story. But in fact, in eukaryotes, you just don't need them. Let me give you an example. So here's an example of the GAL genes that we've studied a lot. And so before you induce, there's an inhibitor actually on GAL4. These nucleosomes that lie in the promoter are actually rather weakly bound compared to other nucleosomes because it's sort of a high AT. But it doesn't matter. The basal level is 0. There's no repressor. There's no, people get confused about glucose, which will never be understood. But under ordinary conditions, the gene is totally off, and these nucleosomes are sitting here. Now, when you activate, which means remove the inhibitor, what you find is that a protein called switch SNF is recruited, and you can watch with this special assay the nucleosomes falling off, and the DNA becomes ever more free of these nucleosomes. Except in the center here, which is another story, but let's leave that aside for the moment. If you mutate switch SNF, you simply don't activate. Actually, you do, but it takes a day instead of 20 minutes. Almost every effect in molecular biology that we ever study, you say it doesn't happen, and it's in the literature and so on. It does happen if you wait long enough. But the whole point of these things is to make it happen. So the obvious point is that these and it's also well known that if you disturb nucleosomes in general, you start to get basal transcription. So it's hard to avoid the conclusion that the random semi-specific, let's call it, or a binding of nucleosomes is repressing the basal level. And that I mentioned, these guys are phased only because there's a barrier here. If there weren't a barrier, and we know that if we take this barrier and put it anywhere else in the genome, we get phased nucleosomes. Sometimes they bind much more tightly and so on. Doesn't matter. It's a completely independent variable. So let's see, did I leave anything out? So for example, then if we want to look in a higher eukaryote, we look at the murine mouse KIT gene, and in myeloid cells, the gene is off. And this large region is covered with nucleosomes, and these have a lower affinity, which may or may not be important, for the promoter, which lies 150 kb away, which is a CPG island, and therefore these nucleosomes are bound very tightly. Now, if you go to mast cells, where the gene is active, these nucleosomes are largely displaced. You get these proteins binding, looping, as I'll show you in a minute, which everyone agrees now, does an amazing thing. It's just like in yeast. It displaces a nucleosome or two and puts in the machinery. And now, by the way, you'll notice that these nucleosomes are seriously phased. That's because of the barrier of the machinery that's been imposed. Before that, they're not phased at all. So it has nothing to do with the sequence. Is that clear? Sidney, is that clear? Do you care? No. Okay. So just to reiterate it, in eukaryotes, removal or inhibition of the activators, whatever they are, suffices to turn a gene totally off, and that's not true in bacteria. So then you say, so what? It's a minor point. Well, yeah, I already mentioned about cooperativity. I don't know why this has come up again, about the nucleosomes. But what is the important point? Oh, yes, I'm now to number 3. That's it. So I've already said cooperativity is expanded thanks to nucleosomes. You don't need repressors, of nucleosomes. And now something that Mike Levine almost said, you now have a different logic. You can have, here's an enhancer that works from a distance. It loops over and activates this gene. So how do you turn that off? Well, you turn it off by decommissioning, as we say, this enhancer. You take away these activating proteins. So now what happens? Well, the gene goes off, but it can be activated by proteins bound to any other enhancer. If you did it the way bacteria did it and put a repressor here, you couldn't activate from any of these, from the other guys. So this is the secret that enables eukaryotes to use multiple alternate enhancers to activate genes, which modestly put is the secret to life. Does anyone object to that? Is it clear? And I'll just show you an example or two. So here, for example, are the mast cells, and this is the KIT gene. And here you see a huge peak of polymerase. The gene is on like crazy, and it's got these huge, this is called a super enhancer, which doesn't mean anything. There are 4 peaks of a combination of 3 things. There's RNA polymerase in every peak. There's in 2 different histone modifications. Frankly, I always forget what they are because there's absolutely no understanding. Oh, this is also GATA2. All right. But you can measure these, you can detect enhancers by the coincidence of Pol2 and these marks. And, need I say it, we have absolutely no idea what the marks do. Some people claim the marks come in after transcription has begun. They're little transcription of these guys. Now let's go to GIST, which is a tumor, gastrointestinal stromal tumor. And of course, the mast cell enhancers are totally off. And now there's a new enhancer here upstream. This one has 3 peaks, and the gene is on, but not quite as much. So in the tumor, you're activating from a different set of cells and different set of enhancers. And in myeloid cells, where the gene is totally off, there just aren't any enhancers. Negative effect, you don't need negative effects if you got rid of the... of the activators, okay? So then, just to make the point with a slight something else, is that the way the enhancers communicate with the promoters, as it happens in lambda, is by DNA looping. So, but there's an added something here. So here we go back to the mast cells with these 4 peaks of Pol2 and so on, and the gene is on like gangbusters. Now, what if we add shRNA to RAD21? Well, transcription goes down a lot, and confusingly, these enhancer peaks go down. And this and many other suggestive experiments indicate that these guys are somehow talking to each other. Notice what happens in between. Here and here and here and here. We don't really understand that, but it's probably there's some cooperativity involved in formation of these enhancer peaks. And you notice that transcription has gone down. Now, I'll just say one other thing in case I don't get to it. Am I out of time yet? Okay. One, it doesn't look like so much on this slide, but I just want to say it. So when you go from on to less on, if you go from on to off, there's nothing, as I showed you. If you go from on to less on, here are the polycomb marks suddenly appearing. So, okay, I'm going to come back to that. Just ask me about it, because it's easy to make enhancers. So, for example, in doing Yamanaka reprogramming experiments, we compare SOX versus SOX VP16, which is SOX fused to an activating region. And what's amusing is you can make new enhancers that never existed, and they're higher peaks when you have SOX VP16, the stronger activator, than when you have that. And then I could go through a whole bunch of stuff where you eliminate, you make super enhancers, you can destroy super enhancers, and so on. So that's how you regulate genes. One way or another, you inhibit the activators. It's very easy to make enhancers. Of course, there are a lot of details we don't understand, but you don't do it by repressing the gene. That's what makes everything possible. Now, so here's the big picture, and some of you have heard me rant about this before. None of this is surprising. Nothing I'm saying about, well, I got to be a little careful. And I think this makes the role of nucleosomes I've been discussing, puts it in a context. So first of all, obviously, the term gene regulation is silly. Genes aren't regulated. What's regulated is RNA polymerase, so that it goes to one gene versus another. And then when you think about it that way, you realize that almost all enzymes, non-classical enzymes, enzymes that are not involved in seeing small molecules, but rather other large molecules, are regulated this way. Regulated meaning substrate choice is determined by recruitment. So the famous example I always like to use is E2, which has a lot of possible substrates. So just notice the enzyme is constitutively active, and this is true for a lot of these enzymes. Secondly, there's a certain natural tendency to work everywhere as part of the background. That's a problem. That's what nucleosomes help us with in transcription. There's another way of helping us here. The way you get specificity is, of course, by recruitment. You use these little things called E3s, which are entirely equivalent to transcriptional activators. They simply bind to the enzyme and to the specific target. This one binds to the green target, as any fool can see. And I just want to emphasize that as one looks into this, you see that every single enzyme, there are these different and sometimes quite unexpected ways that nature has fought the background problem. So in ubiquination, I'm told that if you do just a little chain, it doesn't do anything. It gets erased by. If you do a big chain, then whatever. There's an amazing story about kinases, and of course there are then you get deubiquitinated. There, of course, are phosphatases that counter kinases, but there are more sophisticated ways, always so that when you get a little bit of it, you you erase it. Always fighting against the basal level, because all these enzymes are constitutively active, and it's just a question of mass action, who they're going to work on. It's amazingly dumb way to run the universe, you would think, if you were to design it, but that's the way it works. The amazing thing is not aging. The amazing thing is that it all works in the first place. And for example, one of the things that can go wrong, and my cancer pals are not impressed ever by this, except they keep discovering it, that if you overproduce anything, you overproduce an enzyme, a substrate, or a recruiter, any of these things, you will tend not merely to turn up a pathway, you will tend to lose specificity. So for example, this is appearing all the time now. Sawyers at Sloan found the amazing thing that the androgen receptor and prostate cancer and so on. So he kept developing these inhibitors. And he more and more finally completely wipes out the tumor, so that because the androgen receptor wants to bind to these DNA binding sites and activate certain bad genes. But he always ends up with resistance. And what are the resistance? They're overproductions of the glucocorticoid receptor, which now binds to the androgen binding sites. You see this with kinase. I have an amazing thing about the PI3 kinase pathway, where they get resistance and blah, blah, and it's all there, and then boom. And what has happened is that now you're overproducing some other kinase, which of course, since it's just a matter of binding, a factor of 10 or whatever, it goes to the wrong substrate. So they always say, well, we're activating this pathway. Yeah, but you see what I mean? In other words, the way the whole thing works, you've got to maintain specificity of these inherently not very specific enzymes. They all have the active site, same active site after all. Like kinases, for example. Phospholipid, you get the idea? And so this makes the cancer thing even more depressing. How are we doing? Well, here, or less. Okay, I was going to make a nod. Why don't you ask me if you want to hear about this? Otherwise, I'll just skip it. Thank you.
I have two comments. In the initial part of your talk, and maybe throughout, you talked about the need for just an amino acid or two to touch another protein, and that was sufficient. But let me point out to you, those couple amino acids are in an architecture which is specifically arranged, and they're in three-dimensional space. So it's far more complex than the simplicity. The simple, it's really complex.
Wait, before you go too far. The first real demonstration of this we had was a mutation in a member of the, what's it called, Roger? The mediator, called GAL11. And it had acquired a mutation that now did an interaction it wasn't supposed to do. Okay? It's a very, so now you take just a small sliver of that protein, stick it onto any other protein, and it works. So the only problem is to expose these, as far as I know.
Well, it's a question of presentation. It's being presented by another protein. So that's my point. Now let me get on. This is a teleological question, or it's a point. It's not for you. I hope there's people in the audience that have thought about this, but it just so happens the diameter of an alpha helix is the same as the diameter of the major groove in DNA. So the question is, how many other protein motifs were explored before nature settled on the alpha helix? Is anyone studying? Do you know? Is that?
Oh yeah, there are millions of.
Well, I know these other motifs, but ones that have this regulatory ability to bind DNA specifically.
Yeah, there are millions of motifs, and not all of them fit this simple, like the homeobox and like lambda repressor helix turn helix. Yeah. Yeah.
There are multiple ways of binding the major group DNA to touch the bases to discover the sequence. So it turned out that way with the alpha helix, but I doubt if they were correlated. I think it's unlikely that those two events happened together, forming DNA and alpha helices. They were not coordinated. I mean, the implication of what you said.
The other, obviously.
Yes. But I'm saying that proteins found plenty of other ways of doing it. So
I'd say it's an
wasn't
argument,
restricted to
after
that.
all, for intelligent design, because if you're going to make the alpha helix, I mean, and it fits in the major, I always thought it was miraculous.
All intelligent design. There's no
Once that
question.
was, yeah, once the alpha helix was found, it was used repeatedly for many interactions with DNA, genetically once it was found.
But I mean, I don't know. Well, yeah, you're right. So the talons are a magnificent example. They stick a finger in, and it's like zinc fingers and so on. Yeah. Yeah. So maybe life would exist even if it weren't true.
It's interesting that the reason why people missed this whole idea thought that you couldn't read a DNA sequence. You couldn't have a sequence-specific DNA binding protein based on the early knowledge of globular protein structure and the way in which substrates enter a groove in the protein. And therefore, he came up with the idea of separating the strands for recognition so that the bases could enter a groove in the protein.
I never heard that before.
Did he ever
If you look at
publish that?
1970, oh yes, 1971.
I've tried to find
A
it.
general model for the structure of higher organism chromosomes.
Where was it?
Nature.
Because
Nature,
Jim
1971.
Watson
You
told
should
me
read
that.
paper.
I've been looking for it because Jim Watson. I know what the argument is. I just never saw the paper. No, Jim told me that Crick went to bed for 3 weeks when he saw what the real answer was. I'd love to find that paper. Can you send it to me? Great. I've heard about it. I just can't find it.
A general model for the structure of higher organisms.
What he got wrong is exactly what we just discussed.
Any other questions?
I have this great quote from Crick that I used at a college talk recently to try to get some reactions from students. I said, you people are all too passive. You don't want to criticize each other. And I put up a statement, a magnificent statement from Crick saying, if you're going to do science seriously, there is almost no way you will not go down the wrong path, unless you have someone there to tell you, And so then I put up a picture of Wally Gilbert. I said, I found this guy told me I was full of shit every day. And that was incredibly useful, but that's not the culture now, right? And Matt, of course.
We don't even talk because of secrecy between labs.
That's also true.
But science is done with groups of people. It's not done individually, like mathematicians work alone. But science has to be done with groups of people. Really, it is.
Yeah, but there's not. Yeah.
Explain that.
An idea, Mark was talking about ideas, they happen in the minds of individuals.
They happen in the mind of individuals that are influenced by other individuals. What's the general?
Well, if they are. Most students now have never heard of Jacob Monod, so why would they be influenced by any of that?
Because it's in the textbooks.
Oh, come on. I can tell you what's in the textbooks. It's complete, You read about gene regulation now, and it's just like fairyland.
You need more people buying your book. That's what you're doing now.
Actually, there are 2 books, Apparently. Yeah, okay.
An idea starts in one mind, but then you talk about it and it changes and changes. And when they remember it, it was there right in the beginning in my mind, but that's not true.
Well, I've always said I think Tom gets entirely too much credit for all the things he's done after leaving my lab. Do I get any credit for that? No. Forget Ptashne. Never mind. So y'all buying it? No problems? Jesus Christ. Okay.